Yeah, gotta make this song because I need a better mark in bio. So that's why I'm making this song about style theory. And this is public humiliation, but I got it. Yeah, I know sometimes cells can be confusing, right? They got so many parts working through day and night. But listen, I'ma break it down, teach you how they thrive. From a cell theory, you know all life is alive. There's prokaryotic, simple, no nucleus inside, while eukaryotes have nuclear envelopes, fortified with nuclear pores. Let the RNA slide and the cytoplasm keep it moving like a tide. Organelles do their job, got no time to rest. Mitochondria bring the power. ATP, nothing less, because APT is like really good, you know? In the endoplasmic reticulum, proteins get checked. Yeah, if it's rough, we got ribosomes. Yeah. Cells gonna work, they function all day, moving through the membrane in a fluid mosaic way, diffusing through the cell in a hyper or hypopae. Balance the isotonic, keeping the stress away. If it's a plant cell, yeah, plant cell. If it's a plant cell, yeah, the cell wall standing strong, holding shape, trigger pressure, keeping it up all along. Got the chloroplast taking sun and making it green, filling up the plastids, if you know what I mean. Yeah, lysosomes clean it up, breaking down the waste, while vesicles move supplies. Gotta keep up the pace. The cytoskeleton, hold it down, give the shape, and if it's moving, cilia or flagella set the wave. Yeah, cilia and flagella, yeah. Well, cells gonna work, yeah, they function all day, moving through the membrane in a fluid mosaic way, diffusing through the cell in a hyper or hypoplay, balance like isotonic, keep the stress away. The balance is like isotonic because it keeps the stress away. Yeah. Endocytosis, bring it in. Exocytosis, push it out. Phagocytosis, eat it up, no room for doubt. And if it's osmosis, water's flowing through the slide, moving through till the levels coincide. Yeah. So next time, if you're wondering how cells survive, just remember that every piece plays a role in the vibe. From the membrane to the mitochondria, spark, life spill on these cells, every light in the dark. Yeah. Alright, good, nice. Thank you. Hi guys, thanks for watching that. The first part was for my teacher in my class, because like, all jokes aside, like, although I think it is like kind of public humiliating, that was a joke, but I kind of flunked my midterm due to some issues, but it's really not my fault because like I did study a lot. I'm just trying to, like, you know, it's term 2 and Miss Curtin does this thing where if you make a music video to a song, except you change the lyrics to one of her units, she gives you some extra marks. So that's what I did. This is what I've covered, everything on this white list. And for you guys, basically what I'm gonna do now is I'm gonna explain what the song lyrics mean so it could really help you with this unit in Anatomy and Physiology 12. So yeah, let's get into it. Okay, so I wrote a little intro, and I was just telling everyone how like cells can be very confusing, and when you first look at this unit, it is very, very confusing because there's a bunch of steps to the cell, a bunch of things to do, and it can be confusing, right? But we start off with prokaryotics and eukaryotics. So these are the two classes of cells that exist, the prokaryotes and the eukaryotes. Next, we will go over the nuclear pore. So the nuclear pore is kind of like the outer wall of the nucleus. So we know that the nucleus is a large, centrally located organelle surrounded by a nuclear envelope. And this nuclear envelope is a double-layer membrane, two phospholipid bilayers thick. And this is basically like a continuation of the endoplasmic reticulum. So basically this envelope has pores called nuclear pores, that allow selected molecules in and out of the nucleus. That's why when I was talking about the nuclear pores and letting the RNA slide, what these pores actually do is they are the routes which genetic messages RNA pass into the cytoplasm. Organelles are just the contents in the cell, so nucleus, mitochondria, we got, for example, lysosomes, vacuoles, vesicles, Golgi apparatus, etc. Those are all like basically organelles. Now, the mitochondria in the lyrics, mitochondria bring the power, ATP, nothing less. Yeah. So basically, like, the mitochondria is the cell's powerhouse. That's why, mitochondria bring the power. And ATP is adenosine triphosphate. So the mitochondria is actually the largest organelle in an animal cell after the nucleus. So, like, second largest organelle in the cell. And there's two parts to the mitochondria, the matrix and the cristae. The inner is convoluted into shelf-like folds called the cristae. So in the diagram, of the mitochondria, if you see the folds, that's the cristae. And the inner fluid-filled space is called the matrix. Next, I rapped about the endoplasmic reticulum. Like, proteins get checked if it's rough. It got ribosomes. Smooth keeps the rest. So basically, what I mean by that is in the ER, endoplasmic reticulum, ER for short, the RER stands for ribosomal endoplasmic reticulum. And what happens in the ribosome? Well, it's basically like the site of protein synthesis. That's why we call it RER. It's just like a bunch of little like ribosomes. And if there's no RER, it's just ER is just like smooth ER. And the function of the smooth ER is synthesis of lipids. They are actually also used to detoxify drugs and chemicals in the cell. And that takes place in peroxisome vesicles, which are often attached to the smooth ER. Next, we talked about the fluid mosaic model. It's basically the cell membrane, and in the fluid mosaic model, we actually have a hydrophobic tail and a hydrophilic head. We know that hydrophilic means that the substance likes water. So on the outside, the little balls we see, they're the hydrophilic heads, and hydrophobic, like we don't like that, like deep water phobia, that's what I have, but clown phobia, whatever, yeah, we don't like water. That's why the tails are facing inside. It is basically essentially a double phospholipid layer, and proteins are entirely or partly embedded in the phospholipid bilayer, and it's just kind of like scattered everywhere, If you want to learn more about the fluid mosaic model and the cell membrane and the cell wall function, I might drop another video on that, so you can go check that out, but we're just giving like a little introduction to everything. But anyways, in the rap, basically like moving through the membrane in a fluid mosaic way, diffusing through the cell in a hyper or hypo play, balanced like isotonic, we keep the stress away. In this phrase, we were essentially talking about osmosis, which is basically the diffusion of water across a selectively permeable membrane driven by a difference of concentration of solutes on either side of the membrane. So water moves from a higher H2O concentration to a lower H2O concentration until equally distributed. The reason I said balance like isotonic is because it's a part of tonicity, which basically means when placed in a solution, the concentration outside of the cell may be different than inside. And in an isotonic solution, it's equal. The concentration of a solute inside and outside of the cell has to be equal. So for example, 0.9% salt is isotonic with an animal cell. inside and outside the cell, they both have 0.9% salt. Now, this is where some fun and interesting things happen. If it's a plant cell, yeah, the cell will stand in strong, hold in shape, turgor pressure, keep it all along. Got that chloroplast, taking sunlight, making green, filling up the plastids, if you know what I mean. So, very simple in this section of the rap. When H2O, for example, in grocery stores, in your local grocery stores, they like to put water on top of the vegetables, the plants, and the reason is because this is a hypotonic solution, which is also a part of tonicity. Basically, the water will go inside the cell from a lesser concentration of solute inside the solute that needs more water, essentially. So basically, the cell gains water, and in plants, hypotonic solution actually works a lot better because the plant is not at risk of lysis, which is the cell blowing up. And the reason is because, well, plant cells have cell walls. So basically, what it does is the turgor pressure keeps it all up, and basically, like, your plant is erect and it just doesn't burst. So it's pretty lit. And then, like, animal cells, unfortunately, it does not have the wall, so it does burst. Then we talk about the chloroplast and plastids. Basically, they're only found in plant cells, and the membrane-bound structures that usually contain pigments and give plant cells their colors. The most prominent plastid is the chloroplast, and some plastids are storage bodies for starch, proteins, and oils. The chloroplast is a double membrane-bound organelle in which photosynthesis happens, like the conversion of light to energy. And basically, the chlorophyll is the chemical that absorbs the energy of the sun to provide the energy required for reducing CO2 to glucose. And the opposite process of this is cellular respiration. Next, we talk about lysosomes, clean it up, breaking down the waste, while vesicles move supplies. Well, this is actually very simple. Lysosomes in the cell are known as cellular stomachs. The special vesicles, which scientists used to think is only found in animal cells, are actually also found in plant cells. And they're formed by the Golgi apparatus, but they basically contain a powerful hydraulic enzyme that functions in 1. Cellular digestion, 2. Autodigestion or disposal of damaged cell components like mitochondria, or the breakdown of a whole cell by releasing contents into the cytoplasm. And we call this suicide sacs. And essentially, what the vesicles do is they're formed, vacuoles and vesicles are formed by pinching off from the Golgi apparatus. And the Golgi apparatus is responsible for packaging and modifying the ERs, the endoplasmic reticulum. But basically, they are used for transport and storage of materials. Vesicles are used for transport and vacuoles are used for storage. But basically, the vesicles, since it is used for transport, that's why I said, while vesicles move supplies, gotta keep up the pace. And the cytoskeleton holds it down. In the rap lyrics, the cilia and flagella set the wave. It's because the cilia and flagella are essentially like the movement of the cell. That's how the cell moves. In our bridge part, we talk about endocytosis, bring it all in, exocytosis, push it all out, phagocytosis, eat it up, no room for doubt if it's osmosis. We talked about osmosis, and basically endocytosis means absorbing things from the cell membrane, and exocytosis is excreting it, pushing it out. And when endocytosis happen, 2 things can happen: phagocytosis and pinocytosis. Phagocytosis is cell eating and pino is cell drinking. Cell drinking, absorbing a bunch of little things, and phagocytosis is absorbing one big thing or multiple big things instead of a bunch of little small things.
Hi guys, it's 2 a.m. and I didn't sleep for like 3 days, but that was it of the video. I hope that helped you guys. Thanks.